So, the topic that we are going to study today is about energy of activation and temperature affecting enzyme activity. These two subtopics are from the chapter enzymes. So, in this whole lecture, we will going to focus on the activation energy of enzymes and how the activity of enzymes will affect by temperature. So, these two topics are there in the chapter enzyme. So, okay. Activation, energy of activation, right? So activation, we can also say energy of activation, or we can also say activation energy. So both are correct terms, right? So see, if a reaction is happening in our body or outside the body, that reaction requires specific amount of energy, right? So this is the amount of energy which should be added in that particular reaction so that the molecules inside that reaction should cause a motion, a particular motion will be caused, and that will proceed the whole reaction to occur in a proper way, or you can say the reaction will be with its high speed, right? So that is the reason energy is required to start the reaction. Okay, whenever we talk about a known living system means we are talking about the system or the environment which is outside, okay, outside the body. So when the system is known living, like we have known living system, what we use as an energy source is we use heat as an energy source, where heat is the source which will increase the number of effective collisions between the molecules of that particular reaction which is happening outside the body, right? So in alternative to this process which is happening in non living system, in living systems what we do is we do not give heat as an energy source, right? We do not give heat, cannot be used an energy source of activation, right? So let's explore the reason behind why we are not giving heat as an energy source, although both will require energy sources, right? But in different forms, right? So, we will find out what is the reason behind why we do not give heat as an energy source in specifically living systems. And whenever we talk about living systems, we are focusing on our body, living cells, right? So, see, all living cells, right, in our body are temperature sensitive, right? They have sensitive proteins, sensitive proteins in a sense like they are temperature sensitive proteins, right? And if they are temperature sensitive proteins, it means that it will require an optimum temperature, right? An optimum temperature or an optimum energy to regulate that reaction. So that is the reason we do not give energy in the form of heat, although we are giving energy but not in the form of heat, okay? And the another basic reason that we are not giving heat as an energy source is there are thousands of reactions that are happening in our body. So we can't give each and every reaction, a single reaction, heat as a source of energy, right? So that these are two basic reasons that we are not giving heat as an energy source, right? So and the another thing is, and the another basic reason is like living systems will work in isothermal conditions. Isothermal conditions means the living system works best when the temperature is constant. So these three basic reasons are there. We are not giving heat as an energy source, right? So the last segment of this slide is, you can see here like there is a reaction, right? And this reaction is showing enzyme activity, right? So this E actually depicts enzyme, right? This E is enzyme, okay? This E is an enzyme here, okay? This E is enzyme, and this whole reaction is showing the mechanism of enzyme activity, right? That how exactly enzymes will going to bind with reactants. So this A is showing reactant. Okay, and this is reactant basically. Okay, so A is reactant. So see if this is the side where we have reactant and enzyme, and this is reactant. So let's see how they will going to join together and how they will react in a particular reaction. Okay, so this is actually a reactant. See, enzyme is there and there is a reactant. Let's suppose there is a reactant A. So what will enzyme do is it will combine with reactant A. You can see it has been making a complex, a transition complex, in which enzyme is going to bind with complex. Uh, Enzyme will going to bind with a reactant, okay, and it will finally form a complex. Okay, it will be intermediate. Okay, this is intermediate. Okay, this is intermediate complex, and this intermediate complex is another reactant. Okay, this is intermediate complex, intermediate. Okay, so this is intermediate complex, and this intermediate complex will again change. Okay, the reaction is still in continuity, where you can see here B is also there. It means B is another reactant. A was there, reactant. Okay, and this is the second reactant we have here. Okay, so what will happen that the enzyme will going to bind this A reactant and B reactant in continuity, and enzyme will isolate when the product will form. Okay, so this is how enzyme react in a particular reaction. Okay, so let's proceed toward the graph. Okay, so this graph is showing the activation energy. Okay, um, actually, the significance of giving energy is, we have already discussed that this energy is going to help us in a chemical reaction. Okay, activation energy or energy of activation. How it will going to break the existing bonds. Okay, it will going to break the existing bonds. Okay, and the reaction will continue. Okay, but there could be another qualitative process. How when this reaction in while this reaction is happening, we can also put an enzyme here, and this enzyme will help us by reducing this activation energy, right? Although we have to give the energy, right, but this enzyme will help us to reduce this activation energy, so that in a minimum amount of energy the reaction will proceed. So that is why what we are doing, that what we are doing is like we are putting enzyme, okay, enzyme in that particular reaction, so that the activation energy should be decreased, or we can have the input of energy in a lesser level. Okay, so that is why enzyme has greatly influenced the reaction. Okay, so that is why let's see in this particular graph, that what happens if there is enzyme, in the presence of enzyme and in the absence of enzyme, what will be the graph. See, and this blue line is showing the presence of enzymes, and in this particular place you can see that the particular energy is utilized in less amount as compared to the reaction which has not enzyme. So see, here is uncatalyzed reaction pathway. This terminology says uncatalyzed reaction pathway. Uncatalyzed reaction pathway means there is no enzyme. Okay, so see, we have already discussed in our previous lectures that catalyze we have said in our enzymes definition that enzyme is a catalyst, right? Catalyst means it is speeding up the reaction, right? So if we experiment a reaction without enzyme, okay, so here there is no enzyme, right? Here there is no enzyme. In this condition, what happens that we will require to put more activation energy or energy of activation. So this graph actually depicts the same thing, that energy of activation is required more in this case, right? But when the enzyme will absent, the reaction will proceed with less energy. Okay, so when you will proceed this towards this part, okay, toward this part, okay, this part, you can see that the energy is required in less amount, right? So it's because activation energy is happening with enzyme, right? So that is the reason. Here are our reactants, as we have discussed in the previous slide. We have reactant A, and there was an enzyme, okay, and they had made a complex, right? So we have product here. Product means the substrate will going to convert into the product. So this is how the reaction will proceed from reactant to the product. So this is x-axis where they have shown reaction process from reactant to the product. So this is how enzymes work with activation energy, right? And it reduces the activation energy basically. And this graph actually shows us the same thing, that the activation energy is reduced due to the presence of enzymes, and when it is absent, then we will require loads of energy inputs, right? So the next thing is about temperature. As the topic depicts in the previous slide, that we are focusing on, the first thing was about activation energy or energy of activation, and the another thing is about temperature. So let's see the enzyme activity is affected by lots of factors, and in this particular topic we are focusing on temperature. Okay, so we will see actually that how exactly temperature affects the enzyme activity, like what happens if we increase the temperature. Okay, what happens if we increase the temperature from normal, what happens if we decrease the temperature from normal, what will be the enzyme activity. So we are focusing on the temperature as a factor, right? So see, before starting with this, lowering temperature or increasing the temperature, we will first analyze the importance of temperature, right? So see, like whenever the reaction is happening, we have to give the temperature. Okay, we have to give the energy. Okay, so see, heat we have already discussed actually. See, what was the heat doing in a reaction is it actually speeds the molecular motion, like it was have an integral role in doing collision, okay, effective collision within the molecules. So that is the reason we are giving heat. And it what does the heat doing is, like, heat is actually proceeding the reaction through providing motion to the molecules, right? So see, whenever we say molecules, we are actually talking about the substrate. Okay, so we have a substrate. This is the particular molecule on which an enzyme actually acts, right? So the molecules of substrate will going to move. Okay, they will be in motion and they will move quickly at a speed or rate, right? So this is how the reaction will increase its pace, increase its speed, right? So okay, and the another thing is, like, whenever we increase the speed, it will actually increase the rate of reaction. See, what exactly this statement says is, like, an increase of just 10 degrees centigrade in the existing temperature doubles the rate of reaction, but to a certain limit. So what does this line or this statement means is, like, whenever we increase the speed up to 10 degrees centigrade, just 10 degrees centigrade, what will it happens? Okay, what will be the resultant of this 10 degrees centigrade increase in temperature is, like, it will double the reaction, it will increase the reaction. Why is this speed it is because we have increased the temperature. So degree centigrade is depicting the temperature. So whenever we increase the temperature, the reaction will increase. But see, there should be at a certain level. Okay. um, See, I will erase this part to show you the certain limit here. Okay. Okay, so it is not erasing actually. So um okay, see, uh this is but to a certain limit. Okay, so this line is but to a certain limit. So it means like we can do like we are just increasing the temperature and increasing the temperature and increasing the temperature, and we are saying the rate of reaction will increase. No, it does not happen in this way. We have to give the temperature but in a limited way. Okay, so that is the reason this line says that but to a certain limit. So we have to give the temperature but to a certain limit. Okay, so we will actually discuss further that why we are saying certain limit. Okay, so the another terminology, the another basic concept that we will discuss is about optimum temperature. Okay, optimum temperature. Okay, so see, the temperature which we are giving to the reactions, to the chemical reaction, the temperature should be optimum. And what does exactly optimum temperature means is like the temperature which will work best for the enzymes. Like see, temperature that promotes the maximum activity. Okay, we have to select the temperature which will going to affect the enzyme activity in a better way, right? So the temperature will promote, so this is the temperature which will going to promote the enzyme activity. This temperature is not declining the enzyme activity, but it is promoting the enzyme activities. So that kind of specific temperature is called optimum temperature, which will promote the enzyme activity, the action of enzyme or the chemical reaction. Okay, but to a certain limit again. So this statement also says the same thing, that temperature increase. Okay, increase what will happen if you increase the temperature, the reaction will increase, right? The rate of reaction will increase. But above this level means like if we are giving temperature above this optimum level. Okay, this level means we are talking about that optimum temperature. Okay, this optimum temperature, if we give above optimum temperature, this arrow actually. Okay, above optimum temperature, what will happen is like as we have discussed before, that we have certain proteins or which are temperature sensitive. Okay, they will disorient if we will give the temperature. Okay, without keeping in mind about its optimum temperature. So what we have to do is like we have to see the level. Okay, optimum level or optimum temperature. Okay, if it increase or decrease from this optimum level, the reaction will not occur in a better way, or it will not promote. Okay, see, if the reaction is increasing from this optimum level, the secondary and tertiary structure of enzyme will disrupt. Okay, as we have already said. Okay, as we have already said, that the reaction will disrupt. Okay, and the molecules are temperature sensitive. Okay, so that is the reason what we have to do is we have to give the energy, we have to give the temperature which will be at optimum level. Okay, if we are not giving the temperature at optimum level, what will happen, the resultant situation will be um, the secondary and tertiary structures will be disrupted. Okay, so these are the actually structures of enzymes. Okay, secondary and tertiary structure, like we have different chains, which are arranged in complex form within the enzyme, so they will be disrupted because we are giving energy, not in a limited way, not in an optimum way, but it has increased its limit actually. Okay, so the next thing that we will discuss is we will analyze the difference between maximum temperature and minimum temperature. Okay, in the previous slide we have discussed about optimum temperature, so let's explore the concepts behind maximum temperature and minimum temperature. Okay, so we will discuss maximum temperature and minimum temperature. Okay, see, whenever we say maximum temperature, we are talking about the temperature which has denatured the enzyme. Okay, so this temperature is actually the temperature which has crossed the limit from optimum temperature. Okay, optimum temperature. If this is the optimum temperature, okay, if this is the optimum temperature has crossed the limit and the enzymes has denatured. Okay, so the optimum temperature will cross the limit through increase in its temperature, right? So the temperature which causes denaturation of an enzyme is called maximum temperature. Okay, so this is very easy concept that when the temperature will exceed from optimum temperature, then that temperature will called as maximum temperature. Okay, so, see, the another reason why the enzymes denature or their actual shape disrupted is because the hydrogen bonds within that particular structure, like we have talked about the structures like tertiary structure. Okay, we have discussed about secondary structures. So within these structures, there are chains, and these chains and these molecules are bounded by hydrogen bonds, and because of this higher limit of temperature, it actually breaks the hydrogen bonds. Okay, so the hydrogen bonds are they break. Okay, and if hydrogen bonds break, then of course the appropriate structure of secondary and tertiary will going to disrupt. Okay, so we must also keep in mind that hydrogen bonds are temperature sensitive. As we have discussed, proteins are temperature sensitive. In the same way, hydrogen bonds are also temperature sensitive. Okay, and the another thing is, and the another concept is about minimum temperature. Okay, see, in this case, in this particular case, what we are doing, minimum is showing that we are reducing the temperature. Okay, reducing the temperature. Okay, we will reduce the temperature up to the freezing point or even below from this freezing point. Okay, below from this freezing point, we what we will do is like we will decrease the temperature. Okay, we will decrease the temperature. Okay, let me erase this. Okay, so I will pick my pen as well from here. Okay, so what we are doing in minimum temperature, we are decreasing the temperature. Okay, we are decreasing the temperature up to the freezing point. Okay, and we can also decrease the temperature from this particular freezing point as well. Okay, so see, like there is a question, what will happen if we are decreasing the temperature continuously, right? What will happen? Okay, the enzyme will inactivate. Okay, the enzyme will inactivate. So the way they were active in optimum temperature they will actually they will not work in a proper way because their activity in active form has been converted into inactive form. Because why is it happening? Because we have decreased the temperature from its optimum value. Okay, in this case the enzymes are not denatured. Enzymes will denature if we are giving temperature above its optimum value. When we are giving maximum temperature, the enzyme will denature. See, denature. But if we decrease the temperature, if we are given minimum temperature, it means the enzyme will be inactive. Okay, the enzyme will be in the inactive form, they are not denatured. Okay, and the best thing about inactive enzyme in this minimum temperature is that we can regain this, we can regain the process of inactivity of enzyme into active form. Okay, so that is why here you can see that regain their catalytic influence. How exactly it will happen is like we will increase the temperature gradually, and whenever we will increase the temperature gradually, what will happen is like the catalytic activity will restored again. Okay, the enzyme will again work the way it was working before, right? So in this way, the enzyme will work better because we have given the optimum temperature again. Okay, we have given the optimum temperature again. That is the reason the enzyme will work better and it will restore its higher temperature. Okay, so this whole process, right, where enzymes are in inactivated form and they are, again they are becoming active. So this whole process is depicting two forms. Okay, in the first phase, in the temperature is decreasing. Okay, gradually the temperature is decreasing, and again we are, changing that form, the inactive form, into the active form actually. And whenever we will change, we will shuffle through increasing the temperature, what will happen is like it will increase, it will regain its catalytic activity. Okay, catalytic activity. So this whole process actually is about minimum temperature. Okay, where what is happening, uh, where inactive enzymes become active again. Okay, the temperature which we give to the inactive enzymes to make them active again is called minimum temperature. Okay, so this is the particular temperature which we are giving to the inactive enzymes. First we will have to inactive make the enzymes inactive by decreasing the temperature gradually. Okay, so the next thing is, okay, so this is the last slide of our today's lecture, where we have two graphs. Okay, these both graphs are showing temperature. Okay, like we will see how exactly enzymes work. Okay, and this graph will help us to make us understand about the enzyme activity. Whenever we increase the temperature, what will going to happen, and whenever we decrease the temperature, what will going to happen. Okay, see, this is rate of reaction. It means like the reaction, the rate of reaction, the speed of reaction. Okay, so this is the temperature. Okay, see, if we are giving temperature to enzymes, it is showing kinetic energy. More kinetic energy means like the molecules within this chemical reaction, there will be reactants, there will be substrate, they will collide with each other. And whenever we give its optimum temperature means like humans optimum temperature is about 37 degrees centigrade, right? So if we are giving 37 degrees centigrade to enzymes, okay, so they will work best. Okay, so this slope actually showing that the work is best. Okay, like the enzymes are working at best. This is the particular temperature, optimum temperature where enzyme work best. Okay, but when we increase the temperature from this optimum temperature, C37, whenever you see here, when proceed this line, you will see, okay, you will see that the temperature is increasing. Let's suppose here the temperature is 39 degrees centigrade. Okay, we have increased the temperature from optimum temperature, like we have given a maximum temperature. Okay, so in this case, the enzymes will start denature. They will disorient the tertiary structure, and the secondary structure will disorient. The hydrogen bonds will disorient, okay, because they are temperature sensitive. So this is how this graph is depicting about the enzyme action according to the temperature. So this is the best temperature, this is the gradual temperature that we are giving, and the enzyme activity is, proceeding upward. But when we will give the temperature which is more than optimum temperature, then the enzyme will denature. Okay, see, in the next graph we have two graphs. Okay, one is showing the enzyme activity on 37 degrees centigrade. This is the same graph as is here. Okay, and the another graph is about thermophilic bacteria. Okay, okay, thermophilic bacteria. Okay, um, thermophilic is hiding. Okay, so let me erase this. Okay, thermophilic bacteria. Okay, so it, okay, now we have already understood about this enzyme activity in human that is occurring at 37 degrees centigrade, but we will focus on a new information about enzymes which are in bacteria, specifically in thermophilic bacteria. See, whenever we say thermophilic, it means these organisms or these bacteria are heat tolerant. They have the capability to tolerate more and more temperature. Okay, it is even from above 70 degrees centigrade. So this will have a slightly different graph, because the reason is, like, see, if the temperature was here, the optimum was about 37 degrees centigrade, right? But we will increase the temperature for this bacteria, because it has the tolerance over increase in temperature, so it will work best on 70 degrees centigrade. Okay, although our human being, the enzymes of human being will denature, but the bacteria or thermophilic bacteria has the ability to tolerate the heat. So it the enzymes of this bacteria have the capacity of tolerating the heat. Okay, so that is the reason they will work best on 70 degrees centigrade or even 77 degrees centigrade. Okay, so it means we have learned another new concept, that there can be enzymes, okay, which can be heat resistant, and they are specifically present in bacteria. So this is the end. Thank you so much. Do subscribe and like and watch the whole video. Thank you so much.